Welcome to Chapter 18, Peptides and Proteins, Part 2. So in this part, we're going to be looking at the structure of these proteins. So let's get started with our protein structure. So the structure of proteins is understood in terms of 4 levels of organization. So we have primary level, the secondary level, the tertiary level, and the quaternary level. So we're going to be looking at each of these during this lecture. So the primary structure of the protein is just the order of amino acid residues in a peptide or protein. So that is what we looked at actually last part 1, is just the order of these amino acids. So this order we said matters. We would not say that edit, so if we said each one of these is an amino acid, an E, D, I, and a T, we know the word edit does not equal the word diet, does not equal the word tide, does not equal this word tide. So just like the spelling of these letters makes a difference, the order of our amino acids will also make a difference. So every protein is unique based off of this order, and we're going to call this the primary protein structure. So what we want to get out of this primary structure is that it's the linear sequence. So if I have the ASP, ARG, VAL, TIR, these are all our amino acids, and this is one particular order of amino acid. So this is one primary structure of a protein. We still have our N-terminus end and our C-terminus end as well, our terminal end. So it's just the linear structure of these proteins. So different proteins typically contain from 40 to 4,000 amino acids. And there are about 400 distinct dipeptides. So dipeptides mean literally there's just 2 amino acids that are put together. So if there's 400 distinct of them, and that means 20 raised to the second. So 20 is how many amino acids that we had, and just as a dipeptide, so 20 squared. So 400 distinct dipeptides. And if we want to do a tripeptide, so 3 amino acids in a row, 20 cubed is 8,000. For a 100 amino acid chain, it's 20 to 100 is 1.27 times 10 to the 130th protein possibilities. So there are a lot of different possibilities for proteins just by because the order matters. Even going 1 to 2 is different than 2 to 1. I can't say it's a valine then a glycine and then a glycine to a valine. Those are 2 different amino acids. So because we have a C-terminus and an N-terminus. So the order matters, which is why we can have so many different possibilities for this primary structure. Alright, so once we have that primary linear sequence of amino acids, then that sequence is going to start interacting with each other. So depending on these interactions, we're going to get our secondary protein structure based on how they fold, twist, or bend to form or to start to fold into our proteins. So there are 2 types of secondary structures that we are going to be looking at, and these are our alpha helixes and our beta sheets. These secondary structures result from our hydrogen bonding that takes place between our NH and the amide and the C double bond O groups from the different parts of the peptide chain or the polypeptide chain. So we've got lots of different hydrogen bonding possibilities since we have so many nitrogens and oxygens. So an alpha helix is a single protein chain coiled in a spiral with a twist. And for an example, wool. Wool is an example of an alpha helix. So that's our baaing from the sheep. Alright. So these alpha helixes, we said, are coiling into our twists. So we look at each one, these are about 3.6 amino acid residues per turn. So every 3.6, so 3 2/3 amino acids, I'll have one turn of our alpha helix. Now this wasn't a very easy structure to figure out. There was a lot of discussion about what these alpha helixes could look, or what the structure could look like. Then one day Linus Pauling figured out how to do, or how these proteins folded. So I'm going to let Linus Pauling tell you about how he discovered the alpha helix.
One day when I was Eastman professor at Oxford in the spring of 1948 to April 1948, I caught a cold. It was before the vitamin C days. I caught a cold, and after a day or two in bed reading science fiction and detective stories, I got tired of that, and I thought, why don't I discover the alpha helix? Or I thought something like that. Why don't I try to see if I can find how polypeptide chains are folded in a way compatible with all the knowledge that we have about structural chemistry, and such that they can form hydrogen bonds to hold the parts of the molecule together. I took a piece of paper, much like this piece, and I drew on it a representation of an extended polypeptide chain with the distances approximately right and the angles right, except that one angle did not have the right value. I still have that original piece of paper, by the way. This is the bond angle at the alpha carbon atom that didn't have the right value. So I folded the paper. Actually, it took several trials. I folded it along several parallel lines through the successive alpha carbon atoms. And finally, I found a way, by folding the paper, I could make this bond angle have the value 110 degrees. And I finally found a way of folding such that when I put it together, there was a NHCO bond formed by each NH group and each CO group, a hydrogen bond which held the helical structure together and had just the right dimensions. I found that this structure, which turned out, of course, to be the structure of hair and horn and fingernail and muscle, and also present in myoglobin and hemoglobin and other globular proteins, a structure named the alpha helix, had 3 6/10 residues per turn of the helix, a helical structure in which there are 3 6/10 residues per turn.
So, kind of amazing that he was laying there sick in bed and got bored and goes, hey, why don't I try to figure out this structure? So next time you're sick in bed, no excuse not to do chemistry homework. No, I'm just kidding. Not all of us are Linus Pauling and can figure out the alpha helix in our bed while we're sick. But kind of a cool story for him to say how he actually discovered this two-dimensional structure. And kind of cool that he could just do it with paper. He just wrote the linear chain on some paper with the correct bond angles, and it figured out there wasn't a right bond angle, so he made the right bond angle, and it just happened to work out to give him the perfect structure. So that is how we figured out that these coils are our alpha helix. So we're going to talk a lot more about the alpha helix here in a little bit, but let's introduce the second type of secondary structure, and that is our beta sheets. So this term beta sheet, or a beta pleated sheet, refers to a sheet-like arrangement that forms when different segments of polypeptide chains are laid side by side. So in these beta sheets, these kind of rotate back and forth here and interact with each other. So notice that here's our N-terminus and our C-terminus. So here is one amino acid set, and here's another amino acid chain, and another, and another. And then they interact with each other in these pleated manner. So it's kind of a zigzag manner, if you say. This is why you say if something, it's like a pleated skirt or something's pleated, it's kind of that zigzag shape. Now, technically these beta sheets can be parallel or anti-parallel. The one that we saw was anti-parallel because I have an amine group here, so the N-terminus here on this chain and the C-terminus on this chain. But we can also have parallel sheets where we have the N-terminus starting on one side and the C-terminus at the end of both sides. So these can all be parallel, so all of the N's are here and all of the C-terminuses are here, or anti-parallel, which is what we have here. So it's every other is a N or a C. So just like our alpha helixes, these beta sheets are held together by hydrogen bonds. So we still have the nitrogen, the NH, reacting or interacting with the C double bond O of the other chain. So these are all hydrogen bonds that are holding these chains together. So let's look at a few examples of beta sheets and on their macro scale. So one example is silk. Let's take a look a little at silk. So silk mainly contains our glycine and our alanine residues, and silk is flexible but not extendable. So we can move silk. It feels very fluid in our hands, but it's not stretchy at all. It's not stretchy because it doesn't have those coils like an alpha helix. It's all beta sheets that are in silk. So remember, silk comes from these silkworms, so the silkworm is what excretes this protein, essentially, that we make into fabric. And that is why silk is like silky, it feels really fluid in our hands, but you can't pull on silk and it's not stretchy whatsoever, all because of these beta sheets. Alright, next we have our tertiary protein structures. So these tertiary structures refer to the overall three-dimensional shape of the protein, including the folding of the alpha helices and the beta sheets with respect to one another. So if we have a protein, and we have these yellow ones are the alpha or the beta sheets, so it tells you which direction the protein is going. So that's why we have the arrows at the end. And the alpha helices are these pink ones, or the spiraled ones. So notice that we've got lots of this stuff in our tertiary structure. So the secondary structure is just the alpha helices or the beta sheets, but when we put it all together, we're going to get our tertiary structure. So we'll have an alpha helix, and then we'll have a long chain of amino acids, and then we'll get into a beta sheet, and then maybe we'll get into another alpha helix. Just depends on what type of amino acids are in that chain. These amino acids in these long kind of strings don't interact with anything, so they're not going to form those hydrogen bonds with our alpha helices or our beta sheets. So here's a couple more examples. So this one's got a lot of beta sheets and a few alpha helices in purple, but all of the blue ones are the beta sheets. So this is kind of a solid form of it. We know that in each one of these blue rectangles, there's a whole bunch of amino acids chains going back and forth, interacting with one another. And as soon as they stop interacting, I get to these single chained amino acids. So this has got a lot of beta sheets, and this one's got a lot of alpha helixes. So we once again said that each one of these is a secondary structure, and when we put them all together, we get our tertiary structure. So we can kind of pull this apart and get all of our alpha helices in a row. And if we pull those alpha helices apart, then we get our primary structure of just a long chain of amino acids. What's kind of crazy about all of these proteins is that there's a lot of different possible folding patterns for these proteins, because we saw that there's all kinds of nitrogens and carbons that can potentially interact. However, of all of those possibilities, there's only one that we're going to call native or biologically active. There's only one that actually works in our body, and that is what actually forms in our body. Every time we need to make new proteins, that's the one that particularly folds. If it folds any other way, it's not going to be biologically active and it's not going to work in our body. So it's very critical that we get that correct sequence of amino acids, and then that's ultimately going to determine that folding pattern, and then both secondary and tertiary structures are going to depend on that primary structure. So it's very critical we get that correct primary structure, so we can get the correct secondary structure, so we can get the correct tertiary structure, so that it actually works in our body. So let's take actually a look at all of these interactions that we can get to get our tertiary structure. So we can get pockets of hydrophobic interactions. So if we get a bunch of amino acids together that are hydrophobic, so are nonpolar, they can all interact with each other. We could also do our hydrogen bonding between peptides. So we've looked at a lot of the hydrogen bonding. We can also look at the hydrogen bonding between peptide groups and side chains. So this is just between the carbonyl and the NH actually on any of our amino acids. And this is actually between a side chain and part of that carbonyl group on the amino acid. So a lot of different types of hydrogen bonds we can have on here. Our next one is between the hydrogen bonding between two side chains. So we said this was a side chain and the main structure of the amino acid. Now we've got two side chains hydrogen bonding. We can have our salt bridge. Our salt bridge is just an ionic bond. Salt meaning that it's got positive and negative charges. So here's the positive on the amine and the negative on our carbonyl or our carboxylate group. So those positive and negatives are going to interact with each other. Those are called salt bridges. Really, they're just ionic bonds. Another one is a disulfide bond. We once again looked at that in, I believe, chapter 14. Two sulfurs can interact to form a disulfide bond. So if I have a sulfur on one chain and a sulfur on another, they can actually interact and covalently bond to each other. So not only can we have our alpha helices and our beta sheets, but we can have all of these smaller types of interactions as well. So each of these interactions has to be in the correct orientation for it to be biologically active. If anything disrupts these bonds, then it's going to expand out that protein and it's no longer going to work in our body. So the other one was kind of a smaller scale of showing all these interactions. This one is a kind of more of a larger zoomed out view of those structures. So we could have, some chain and, oh, look, those ones interacting. And we go through our chain, oh, look, those interact. And, oh, those interact. Oh, here's another fold, and here's another fold, and here's some more interactions between the chain. So as these proteins are created, as soon as something can interact, they go and form these bonds or form these interactions. And when they do, they fold and bend these tertiary structures. So we talked about, once again, disulfide bonds in chapter 14. And what these disulfide bonds do now in amino acids is that they form a lot of times from our thiol groups on the side chains of like a cysteine. So this SH, when it's oxidized, actually forms or can form the disulfide bond. So this is what happens when you perm your hair. So if you want to go make your hair super curly, well, the way you do it is try to get that tertiary structure of all of these disulfide bonds that are in your hair. Because remember, we just said that all of these proteins are in your collagen, in your hair, in your nails. So when you oxidize this bond, which is what perming your hair does, you're oxidizing these thiol groups. Oxidizing, we said we can remove hydrogens, and we form a disulfide bond. So when you form enough of these, it starts to get a twist into your hair, and that's where you get it curly. Now, if you already have curly hair, you already have a bunch of these disulfide bonds forming naturally in your hair. So when you are trying to straighten your hair, or a lot of those shampoos and stuff that are supposed to make your hair straighter, what they're doing is they're actually reducing these disulfide bonds back to their thiol groups, so back to the normal cysteine, and so breaking the bond, making it straighter. So I know some of you had mentioned in chapter 14 how you were surprised that there was so much chemistry in a salon, but there really is. So that's why, you really do have to go to beauty school to kind of learn all of these tricks. No, they probably don't get down to this level, but this is exactly what's happening when we start messing with or trying to augment our bodies. We do a lot of chemistry to make it happen. Alright, so I missed the last one, but just disulfide bonds are easily reduced to reform those thiol groups, so that's what we were just saying. So this is very easy to go back and forth between these disulfide bonds. Alright, so let's look at our quaternary structures now. So once we get to these large tertiary structures, we then can form a large number of native proteins or combined more than one of these polypeptide chains. So basically we're going to take those tertiary structures and put a bunch of those tertiary structures together to form our quaternary structure. So an example is hemoglobin. Hemoglobin has 4 parts or 4 tertiary structures that come together to form a quaternary structure. So you see the 4 different colors in here, and the red parts are the hydrogen bonds that connect them. So each one of these is a quaternary or a tertiary structure, and we see the secondary structure of the alpha helices. And then if we were to pull it apart, we'd go all the way down to the primary structure of the amino acid chain. So one example of a quaternary structure is collagen. So collagen is actually the most abundant protein in your body, making up about 1/3 of all the proteins in your body. So it can be found in bones, in your skin, in your muscles, in your tendons, in your ligaments, also found in your blood vessels, in your teeth, and even your eyes. What it does is it's kind of the glue of your body. And actually that's exactly where the word came from. It's the Greek word for kolla, which is glue. So collagen is kind of the glue for everything in your body. So provides your structure for your skin and your bones. So and it also is in your cartilage, which is in your joints to kind of help cushion your joints. And the catch is that our body produces this, but as we get older, we don't produce as much or as high quality. So high quality means that it's not folding quite as well as it did when we were young. So that is why there's a lot of collagen supplements. So you can go buy some collagen supplements, and it's supposed to help make your skin firmer and help ease your joint pain, and it's got a lot of different properties. I don't know how effective any of them are, but it's a very popular supplement right now. Anyways, so the structure of collagen is we take these alpha helixes and we put a bunch of those alpha helixes together and create another helix. So each color is a different helix, and we're basically twisting together 3 different sets of helices. And once again, they're all held together by our hydrogen bonds. So just like our secondary and tertiary structures, these quaternary structures are also held together by those non-covalent interactions. So we said they were hydrogen bonding, hydrophobic effect, salt bridge. Everything that we have mentioned in our tertiary is also held in our quaternary structures. And these quaternary structures are important because they cooperate between the subunits is important to protein activity. So this is ATPase, so we're going to look at this a lot when we look at our metabolism. But each one of these colors is a tertiary structure, so it's a separate unit. And they all come together in this ATPase and create ATP. And we're going to see that this rotates, each of these parts rotate and move their electrons around and are able to add a phosphate to our to create our ATP to create energy. So we're going to look at exactly how this works next chapter, but just in case, here's a technical description with these gentlemen in the picture. So this is funny, but it's actually pretty accurate. One thing moves, so this guy in the middle is moving in a circle, and these guys outside move in a certain direction every time the center moves. So this actually is a pretty good human description of how ATPase and how these tertiary structures interact to form the quaternary structure of our ATPase. Alright, so let's see how much of that last section you can remember. In which of the following levels of protein structure can hydrogen bonding play a role? So which ones are held together or can be held together by hydrogen bonds? Well, all of them except for our primary structure. Our primary structure, remember, is our covalent bonds between our C-terminus and our N-terminus ends. So it's the amide bond covalent bond that is held together in our primary structures. Everything else are our intermolecular forces that are holding them together. Alright, so once we know about our quaternary structures, there's two kind of major types of quaternary structures. These are our fibrous and our globular. So the fibrous ones are like our collagen. So this is the protein that exists in long fibers or strings, and include collagen in our skin and keratin, which is in our hair as well. And usually tough and water insoluble, which is a good thing because we don't exactly want our skin or our hair to dissolve in water. Whereas our globular proteins, these are the more spherical shaped, and these are highly folded and do tend to be water soluble. These are the ones that are going to do a lot of the work inside of our body, so they do need to be water soluble. So let's take a look at each one of these in turn. So one of these features of fibrous tissues is that they often have regular repeating structures. So we'll see these regular repeating structures are those kind of spiral shapes. So keratin, which is found in hair, horns, wool, nails, feathers, are these helix shapes. So it's two pairs of alpha helices, and they're going to be wound around each other. And each one has 7 amino acids per repeating structure. So here are those two alpha helices interacting, and then they interact with each other. So this is the actual structure for our hair. So this is kind of the macro scale, and this is the micro scale of it. But let's look even inside of what each one of these hair follicles is going to look like. So if we take a look at this lady's hair, because she looks like she's got some super strong hair for it to get that long. Alright, so we take each strand of hair and we look inside. We're going to see that we've got the cortex around it, our cuticle, and the spindle-shaped cells of the cortex. And we look inside of there, we get our macrofibril, and inside of there, our macrofibril. So the basic building blocks of this keratin then are these protofibrils, helix of our helices. So we have our alpha helixes, and then we have a helix of them. So these two pairs of alpha helices wound around one another. So it is an alpha helix mixed with other alpha helices that are making these long fibrils of all of our hair. So we had mentioned in another lecture about protein buildup in Alzheimer's. These proteins are these protofibrils. These are the long, thin fibrils that are the buildup inside of our brain that is thought to have caused Alzheimer's disease. Say thought to because they're pretty darn sure it is, but it's pretty tough to tell which came first, the Alzheimer's or the protein buildup. But these proteins are always found in brains of Alzheimer's patients. So here's that collagen that we had mentioned. So it's the major protein found in all of our connective tissues. We said in collagen, every third amino acid is glycine. And many of the others are proline. So we either have a ring or a hydrogen, and kind of forms these unique alpha helices. And then those alpha helices then form our fibrils, and then they connect with hydrogen bonds to form our fibers. So we get up to the macro scale at this point. Our protein muscle fibers are also fibrils, or these long chains of protein. So the myosin in fibrinous proteins is our muscle cell. So both the myosin fibrils and the actin fibrils are what make our muscles move, allow us to move. The myosin is it's like a hook and loop structure. So we think of it like Velcro. There's one side that's kind of all of the rough side that has all of the hooks on it, and then there's kind of the fuzzy side where it has all the loops. Well, the myosin in our muscles is all of the hooks, and our actin will be all of the loops. So our muscles actually contract by a hook grabbing onto a loop and moving the muscle. Then when you're done, it releases it and it goes back to its original structure. So it's basically like pulling and pushing Velcro every time that we are moving our muscles. But it's a lot easier to connect and disconnect these structures versus something like Velcro, which is super strong. So here's kind of a look at what those hooks would look like. So we have this long chain of alpha helices, and then we have these heads, which are super strong polar heads. So they interact strongly with other molecules, so I hook onto it, and then it's done, the interaction comes off, and the muscle contracts. Here's just showing some more of those actin bonds. So it hooks on, and then it comes off to form to expand and contract our fibers. So I found a parody song on myofibrils and how muscles actually contract and interact with the actin and the myosin. So we'll watch a little bit of it, at least for the first chorus, and hopefully you enjoy it, and hope it kind of helps you remember how these interact.
Calcium to the cytoplasm in the myofibrils. It will turn troponin on, causing the contraction of the myofibrils. Signal from the brain rides a nerve to the muscle where the myosin inside will slide. That's the acting of the myofibrils.
Alright, so we'll leave it there. We don't need to listen to it as a good guitar solo, although it's very good. Get a little bit, fun way of listening to it, how the myosin and actin are moving back and forth, and it's why we need calcium, and why you get muscle, to help your muscle contractions. So calcium is not only for our bones, but we need it for the ions to send the signals, to get the contractions, to tell the myosin and the actin to interact. Anyway, so it is actually fairly complicated process for our bodies every time we move, but kind of a cool parody for us. Alright, so let's look at some other types of fibrous proteins. So fibrin, which is shown below, is the fibrous protein that makes up silk. So we think of each of the little spindles from a spider forming these long fibrous proteins that we know are made of beta sheets, and we wind them together to make our silk cloths. So silk is also, we said, a fibrous protein of only beta sheets. So it's got a repeating pattern as well. So it's glycine alternating with layers of our alanine and our serine in these beta sheets. So notice that I have this long protein, and then I have a beta sheet of mostly glycine and alanine and serine, and then I'll get a long string, and then I'll have another one, and I'll get a long string and have another one. So we've got mostly beta sheets making up these long fibers of our proteins, so of this silk. So we're actually not as familiar with globular proteins, even though they're super important in our body, because they don't normally exist outside of our body. So these globular proteins, we said, are highly soluble and very diverse spherical proteins. So these are the big globs of proteins that you would think of if you said, oh, what's a protein look like? So our enzymes, our transport proteins, like our hemoglobin, our receptor proteins, are all globular proteins. So this is myoglobin down here. We also looked at hemoglobin before, and we said hemoglobin had 4 hemes, or 4 tertiary structures that combine to form that quaternary structure. So that's a big globular protein. Now, one thing that we didn't have in our fibrous proteins, because they're just long fibers, is binding sites. So long fibers a lot of times are just holding stuff together or moving stuff in a long chain. Where these globular proteins, they're doing stuff. These are our active proteins. So we need some kind of binding site for it to interact with something. So interact with the oxygen, or interact with, if it's in a white blood cell, interact with a virus or something. So it's going to have some kind of binding site on these proteins. So they're going to be, these big globs are going to have certain little inlets that can interact with everything else that it surrounds it. So we're going to look a lot more at enzymes and in our last lecture in part 3. We're going to devote all of part 3 to enzymes. So we're not going to talk too much about them now. I do want to look at is our simple versus these conjugated proteins. So simple proteins just are composed of amino acid residues. So an example of a ribonucleose, which contains 124 amino acid residues. So we can have any one of our structures in our simple proteins. So what makes them simple is that they're only amino acids. So how that compares to is something that, like a conjugated protein, that has what we're going to call a prosthetic group, or something added onto it that's not part of an amino acid. So an example in hemoglobin would be iron. There's iron molecules in hemoglobin that allows the oxygen to actually bind to the hemoglobin. Because the oxygen in itself doesn't necessarily want to bind in anything actually in the amino acids. The amino acids come together and form what we're going to call a heme group. So it forms this kind of chain or circle around an iron. So we ingest the iron, gets into our bloodstream, a hemoglobin picks it up, and then once it's interacted with the iron, it can go and pick up a oxygen molecule. There's also an iron atom or iron in our myoglobin as well as our hemoglobin. In myoglobin, though, it's actually a lot stronger attraction to that oxygen. So the hemoglobin is kind of that transport of the oxygen to get from one place to another, but the myoglobin is where it's actually stored. So the oxygen storage in protein and skeletal muscles. So it's typically transferred from that hemoglobin to the myoglobin since it's got that stronger attraction. Hemoglobin is also much better for transport because there are 4 different heme groups. So we can transport more oxygen. Whereas the oxygen the myoglobin only has one site or one iron that it can bind the oxygen to. So this whole myoglobin has one oxygen that it can store. So it's not as efficient in storage. However, it is a stronger binding, so it binds it better than a hemoglobin would. Now, we said that the myoglobin has a stronger interaction with the oxygen than the hemoglobin, but that's actually a good thing, because when we're transporting something, you don't really want the hemoglobin to hook onto it very tightly. If it does, then it's not going to want to transport it, it's just going to hook onto it itself. The catch to this is that iron also can bond carbon monoxide, also super strong. So it actually binds carbon monoxide stronger than it would oxygen. This is why carbon monoxide poisoning is so deadly. It's because instead of your hemoglobin transporting oxygen like it should, it actually binds to a carbon monoxide. So instead of holding that one oxygen, it holds onto that carbon monoxide and it doesn't let it go. So instead of dumping off an oxygen and going to get more, this carbon monoxide blocks that site. So then we are actually suffocating because we can't transport the oxygen through our body. So it's not that we can't ingest or can't inhale any more oxygen. It's just that it can't get transported any longer because all of our heme groups are hooked up with a carbon monoxide and therefore it can't get transported through the rest of our body. So even for carbon monoxide poisoning, if you were to run outside and try to breathe fresh, clean air, it's not going to help you because your body's still, all the hemoglobin are still hooked up with those carbon monoxides. So you actually have to wait for that hemoglobin to denature and dissociate and your body to form more hemoglobin until that you could start breathing again. So this could take several minutes. So if you can have low oxygen for a little bit, so if you're exposed slightly, that's okay. But if you're exposed to a large amount of carbon monoxide and you have a large amount of carbon monoxide bound to your hemoglobin or to your hemoglobins, to your irons, then you are going to have a long-term oxygen deprivation and it will kill you. A really sad story I heard recently was of a young boy. I think he was between 4 and 6 years old, and he was out boating with his family all day. The catch was he sat next to the engine of the boat. So he sat in the back of the boat right next to the engine all day long with that engine running. Well, engines are notorious for emitting carbon monoxide. So this poor boy, after being exposed to carbon monoxide all day long, his body finally got too much carbon monoxide and he fainted due to a lack of oxygen, fell into the water, and drowned. But they found later on that even if he hadn't drowned, they couldn't have saved him because his body had been deprived of oxygen for too long. So those long exposures of carbon monoxide just bind up all of those hemoglobins and we can't breathe anymore. Which is why we have all of those carbon monoxide detectors in our house now. So if you're required to have a carbon monoxide detector in your every single room. Now, obviously, somebody's not going to come and inspect your house and check and see if you have a carbon monoxide detector, but it's a good idea to have. And if you try to go and sell your house, you can't sell it until you have those carbon monoxide detectors. So it's just like our fire alarms at this point. You have to have a carbon monoxide detector as well as a fire alarm to sell your house. Which is a good thing then, because you don't want that long-term exposure to carbon monoxide since it is such it's so deadly and you can't undo it once it's been connected. Now there are a couple of hemoglobin variations that occur naturally. One of them is in a fetus. Fetuses typically have a slightly greater affinity with oxygen than normal hemoglobin. And another one is the sickle cell anemia. So you think of sickle cell anemia, it's the sickle cell hemoglobin. So what happens is there's going to be a alteration in the hemoglobin shape, or in the red blood cell shape. So it actually deforms that red blood cell from all of the hemoglobin. So it is a denaturization, or a slight different connections in the hemoglobin than normal. And so then you can't bind oxygen as well since it's not in the correct shape. So therefore it doesn't work as well. It doesn't kill you, but it does make your body not work as efficiently as it could. Alright, so the last part of this lecture, we're going to talk about denaturization. We've talked about it a little bit up till now, and it's basically when something comes apart. So denaturization is the change in protein conformation caused by the disruption of the non-covalent forces responsible for maintaining the native conformation. So in other words, all of those secondary, tertiary, and quaternary structures that have all of those or intermolecular forces, non-covalent forces, those all basically come apart, and all of those structures go back to that primary structure, or the inactive form of our proteins. So since we're losing those secondary, tertiary, and quaternary structures, we're losing all of their biological activity. Once we lose those structures are what allowed all of our collagen and all of our proteins to do their tasks. Without it, they're not biologically active. And it's not normally reversible. So the process is reversible only if minor changes in conformation take place. So one of these is like if you don't water your plant and it starts to wilt. Some of the proteins are losing some of their biological activity. So think of all of the collagen like in our bones holding everything together. It loosens up a little bit and the plant starts to wilt. But you give the plant water and it perks right back up again. At least for a lot of plants, as long as they're not too far gone. But it can be reversed, but most of the time it's not reversible. So let's talk about how we can denature these proteins. So one way is through heat. So a weak side chain attraction of the globular proteins are easily broken. We think about something being heated, and it's moving the molecules really, really fast, and so you no longer have those interactions. And it starts to denature. So cooked meat converts some of the insoluble collagens into soluble gelatin. And we'll see also our eggs, it's actually the agitation and the heat goes from that, basically a liquid to a solid. You can't take cooked eggs and uncook them, right? So it's from the denaturing of all of the proteins inside of the eggs that allows it to solidify. Same thing with kind of our bacon. Our bacon kind of shrivels up with heat because a lot of the proteins inside of the bacon are denaturing. So we also said then for our eggs, the mechanical agitation. So if you've ever beat eggs, so if you ever take a whisk or a blender and you beat them, the mechanical agitation, it forms this foam that you get when you have the beating of those eggs. So that is a denaturing of the proteins inside of the eggs to go from that kind of liquid gelatin to this creamy foam kind of that you get with the eggs. Also detergents, so soaps, when even in very low concentrations, can cause denaturization by disrupting the association of those hydrophobic side chains. So remember, we said soaps had a polar end and a nonpolar end. So if the nonpolar end is interacting with the nonpolar side chains, the hydrophobic side chains, well, then it's no longer interacting with itself in the protein. So then it starts to unravel as well. Which can be a good thing if you are trying to get blood out of some kind of clothing. So, you get a cut and you bleed on your shirt. Well, you want to try to disrupt those proteins so that way your detergent can wash it away. Now, it doesn't do it great, as blood is actually pretty hard to get out of clothing, but that's essentially what it's trying to do. So we said detergents can disrupt the nonpolar chains, but organic compounds in other solutions, such like acetone or ethanol, can interfere with the hydrogen bonding and compete for those binding sites. So we think of ethanol, the OH, or acetone, the ketone. Those are polar, so they can interact with those polar sites, and then it's no longer bonding with itself. A pH change is a big one, which is why we can't have our body change pH too much. We said our biological pH is between like 7.3 and 7.4. We don't want to have a big variation in pH because we saw that having a change in pH, it puts on hydrogens or takes off hydrogens. So those side chains in our amino acid residues are disrupted because they now have extra or too few or too many hydrogens. And also those salt bridges. So if it went from having a charge to no charge, well, that no longer has the bond there, so it disrupts those interactions. Lastly, our inorganic salts, sufficient high concentrations of ions can also disrupt those salt bridges. So you put a lot of salt onto something and it can disrupt all of those bonds as well, since they are also charged. Alright, so lots of different ways to denature proteins. We'll talk about this a little bit more with enzymes, but that's all we have for part 2 of our proteins. So we said part 3, we're going to just be looking at enzymes the whole time and the different types of enzymes and their interactions.